Welcome everybody to the webinar. We are very happy to see many people joining us. I'm going to share the screen just to explain you a little about the form of the webinar. As Barbara said, the title is Computational Aspects of Spatial Biology. If you are here, I think it is not necessary to explain to you what spatial biology is. As you know, this is an innovative approach, an innovative technology, allowing us to combine omics data with histology that in the context of the immune model cross-action is especially relevant, I think, because it is a powerful technique to see whether especially the solid tumors responds or not to immunotherapy, to see whether immune cells and epithelial cells, or better said, cancer cells interact. So there are a plethora of spatial techniques, including several omics and also several platforms. So it's impossible for us to cover all this information in 2 hours. So we have selected 2 examples that we think are very relevant in our area. But before, I'm going to explain you a little bit about the format and the agenda of the webinar, because there are 2 slots of 30 minutes, but you are allowed to ask questions or to comment about the talks at the end of the webinar, right? We have to first 2 30 minutes talks, and at the end 30 minutes to ask questions on live, or if you prefer the chat is also okay, right? So I'm going to present the speakers, and the first talk is by Maria and Heath, Maria Mantas and Heath Patterson from Aspects Analytics, that is a company specialized in bioinformatics and machine learning, located in Belgium. And we are going to give us a very, very interesting talk about a technique they have developed to analyze omics data, but in a multi-omics manner. So Maria, let me see the paper. Maria is the bioinformatics project manager at Aspects Analytics. She obtained her PhD in computational comparative genomics at the University of Edinburgh, and is a specialist in big data, analyzing molecular biology, genetics, and pathways. And since she joins to Aspects Analytics in 2022, Dr. Mantas has been involved in spatial multi-omics integration studies. She is going to explain to us better. She is also including very interesting projects, such as the Allen Mouse Brain Atlas. And Heath Patterson completed his PhD in analytical chemistry at the University of Montreal, and his research focused on lipid mass spectrometry and imaging and 3D mass spectrometry imaging bioinformatics. Wow. And after completing his PhD, he moved to Vanderbilt University to develop integration strategies for diverse imaging modalities. He joined Aspects Analytics in 2023 as a director of spatial biology bioinformatics, and is involved also, as Maria, in interesting and important projects such as like HubMap. So this will be our first talk. Maria and Heath will explain their company, their software for spatial multi-omics. And then we will have another 30 minutes talk by Eduardo Aranda, that is a PhD student, actually in my group here in Spain. And he's a pharmacist and a bioinformatician, pursuing a doctoral thesis in oncological research at Institute for Health Research Aragón. He's using spatial transcriptomic techniques to better understand the molecular communication between immune cells and cancer cells in lung metastasis. So I don't know, Maurizio, if you want to add something.
No, just saying that I'm looking forward to those talks, and yeah, inviting everyone to take notes and take the opportunity to be able to discuss with people that have hands-on experience. So we have a lot of time at the end to discuss any topic and hopefully address some of the more burning questions. So yeah, looking forward to it.
Yeah, don't be shy. Okay. So Maria and Heath, the screen is yours.
All right.
Thanks for joining us.
I'll just share my screen. Can everybody see and hear me okay?
Yes.
Great. Okay, so thank you so much for the introduction. And as mentioned, my name is Maria, and I'm going to be talking a little bit about aspects analytics and the work that we do. So what is Aspect Analytics? We are a software development company in the field of spatial multi-omics, and we provide bioinformatics and AI or ML solutions for spatial biology. Our business has two types of branches, so to speak. Our product branch, which is comprised of our cloud-based platform for spatial biology data management and analysis called Weave, and our bioinformatics service branch, where we can help you and your team analyze spatial multi-omics data, including data integration, data-driven QC, and also co-development, where we can help create solutions that are better tailored to you and your needs. Our mission is simple. We want to provide a unified platform solution for spatial biology. We want to enable interdisciplinary collaboration, and ultimately, we want to unlock the full potential of spatial multi-omics. So far, we have a number of partnerships and collaborations with big pharma companies and also CROs and highly accredited universities. So in the context of drug discovery, Aspect Analytics really aims at speeding up the whole scientific and experimental process by narrowing down potentially successful targets, while also making the whole process a lot cheaper. We work across many therapeutic areas, including but not limited to oncology, immunology, and neurology. And we are technology and vendor neutral, meaning that we can work with any spatial modality, from spatial transcriptomics to spatial proteomics, mass spec imaging, from any vendor that you choose. So if you work with spatial multi-omics data, you know that this data often comes into what we call stacks, which you can overlay on top of one another in a sensible format, and that comes with several challenges. For starters, data integration is difficult due to differences in the coordinate system, meaning that data is acquired with different spatial resolutions in different sections, and they also come in different data formats, most of which are proprietary. And because of that, in Weave, we do what we call stack fusion, where we integrate readouts across assays and sections to get one unified data set. And this can really facilitate downstream data analysis and also interactive visualization, enabling what we call true spatial multi-omics. So this is what our platform looks like. Here on the left, you have the various spatial biology modalities that you can upload into Weave, and there you can carry out any primary analysis that you see fit. From there, you can move on to doing your image registrations and attach data fusion. And once that's done, you can do all types of downstream data analysis, including differential expression analysis and also correlation analysis. You can interact with the platform in different ways. So you can use our Weave UI for your data uploads, downloads, visualization, data analysis, and management. And if you have a computational team, you can also use our Python SDK and APIs to program against the platform. We have our very own and intuitive metadata model that can really tie all of your spatial biology data together in a nice intuitive way. And we take security very seriously, as we are now and still the only player in the field who is ISO 27001 certified. So given that this talk is for the European Cooperation in Science and Technology, I thought it would be fit to talk a little bit about spatial biology data analysis at scale. So we all know that for these large collaborations to work, we need several things in place. First of all, we need efficient collaboration, often via a centralized repo with tight feedback loops for data analysis, sharing of data, and also feedback. We need dedicated analysis pipelines, including standardized processes from data QC, provenance, versioning, and progress tracking, and also scalable computations. And very importantly, we also need to have methods in place that allows for data to be reused. We can offer all of that in Weave, and for that, I would like to talk a little bit about a project that we're currently involved in that is called the GlioMatch project. So the GlioMatch is funded by the European Union, and its goal is to accomplish deep spatial multi-omics profiling of glioblastoma and high-grade gliomas. It's an international collaboration involving 14 different partners. It's going to generate a very large amount of data. Around 500 terabytes are expected throughout the project. And it's going to involve different spatial biology modalities from proteomics to transcriptomics, and also other imaging modalities such as histology and MRI. These data are going to be acquired by different people in different places at different time scales. So Weave here is being used as a central repo where all data is hosted. It interacts with different stakeholders from hospitals to core facilities, and it interacts with different users as well, from clinicians to bioinformaticians, always making sure that the highest standards of compliance and security are enforced and upheld. Very specifically for the GlioMatch platform, the platform has 3 different components. So it's going to have a REDCap component that some of you may be familiar with if you're someone who deals with clinical trial data. This is for patient and sample collection via electronic case report forms. So that's one component. There's a second component that is our Weave platform that I'm talking about right now. So that's for data management, visualization, and analysis. And a third component that is the MRI hub, where people are going to be able to upload, download, and visualize MRI data. And all of these 3 components are linked or interconnected within the Weave environment. In terms of data analysis capabilities, you can expect everything from the basic spatial biology tooling, from tissue annotation, image registration, cell segmentation, to the more advanced interactive visualizations and data management, including sharing of data, full traceability, and also standardization and automation of computational pipelines. So I know that this may sound a little bit abstract right now, but I hope that with this next video that I'm going to show and with Heath's presentation next, we can help demystify this platform that we've been talking about. Just before I press play, I want to say that the data set that I'm going to show here today is a mouse brain data set. And we've acquired different modalities. So I'm going to be showing H&E, MSI, the Luna-4 assay COMET, and also cell segmentation masks. And you can see you're going to see one type of the interaction visualizations that we provide within Weave. So hopefully this works. Okay, so here you can see the H&E here, and I'm going to now make this other H&E appear, which they've been co-registered to. This one is post-MSI, and I hope that you can appreciate the quality of registrations. So the MSI data sets and the overlaid COMET assay from Luna-4, from which we did cell segmentation on, which is what it's showing up on the screen right now. It's at full-scale resolution, meaning that you can visualize individual cells. And as I move in to zooming in on this bit of the data set, I hope that you can appreciate that you can see the underlying COMET assay here underneath with the green fluorescence. Now I'm going to show the MSI data that is overlaid. So here you can see both the cell segmentation masks and the MSI pixels underneath. So they're truly fully integrated. With the platform, you can see also that there's adjacent widgets that you can use for downstream analysis and visualization, from choosing peaks or interval of peaks in your MSI spectra to also different cell types here on the based on biomarkers and also clustering. So I hope that with this very quick introduction, that I hope that I've convinced you today that Weave can offer you an end-to-end solution for spatial multi-omics, that we can help with the full, we can help you with the integration and visualization and also management of all spatial biology modalities from any vendor, and that we can help with also with the co-development of any tools or features that are tailored to you and your needs. So thanks so much for listening, and I'll give the word now to Heath.
Okay, I should have my screen shared now. Yeah, so after Maria gives us the introduction here of what Weave is, sort of the baseline of tools, I'm going to give a little bit more detail on the normal flow of data and data processing within the platform. So to begin, obviously one of the most important things is how do you actually get data into this software? It is a cloud platform, so that means that the data that you have locally stored has to move into our cloud system. We offer a lot of different ways of doing that. So we have a web UI, which works great for some smaller modalities. The Python SDK, which was previously mentioned, where you can script and upload raw data. There's also, we have some upload utilities like command line tools, as well as if we set up permissions correctly on an Amazon S3 bucket, we can just directly access that bucket and move things onto our platform in a piecewise way. Raw data that goes onto our platform, for most vendors, we support just taking the vendor-specific format. So if it's Xenium or nanostring data, we can just take what they output and ingest that onto our platform. And typically what happens is that, you know, we have the raw data, we have some information about the sample, and with our ingestion process, we convert that to our internal formats, which are optimized for cloud visualization and data access through the cloud rather than, you know, access from your hard disk. Importantly, just very briefly, the Weave data model is single section measurements. So if you have an assay where you've measured 10 things on one slide, in the Weave model, we're able to split up all of those different measurements and really make sure that each data bin, each data set is its own entity within the platform. And that ties into how we manage metadata data around each of the all of the data we bring into Weave. So we right now have 5 levels of metadata, and these really capture information about the samples, information about the preparation of the data, all the way down to, you know, things like consumables, date, time, what was the instrument. So just to briefly talk about those 5 levels, they start with donor. So this is pretty obvious. This is like the human being, the animal, cell line, whatever it may be, where your data comes from. And each of these can have their own sort of extended fields of metadata. So, you know, sex, disease, whatever it may be, age is an obvious one at the donor level. The sample is kind of like what we've taken from the donor, or what has been taken from the donor to sample for analysis. So this could be something like a tissue block. In the case of a lot of spatial biology projects, you take a tissue block, you make sections from it. So that's typically what's held at the sample level. Then the sections are the individual, you know, oftentimes we're working with spatial biology data sets, so it's tissue sections that are cut from a tissue block. All of that's captured there. And then we have the assay, which captures vendor information, what was the target list. This is like if you've developed a panel of 40 immune markers, that panel has a unique assay identifier in the system. And acquisition is like a lower level of that tells you, you know, I use this set panel, but I did it on this instrument with these set of consumables that I got on this date. And so once we have all of this, these can all filter into analyses, basically. And in some case, that's obviously required. So if you're doing disease comparisons, you obviously need that metadata somewhere. But it can also be used for useful checks of system functionality. So for instance, QC over time of instrumentation is one area where this can also be used. The next level of management before we get into, you know, of really spatial multi-omics studies is that what we see is oftentimes there will be a given sample or a set of samples from that researchers have collected. And typically they plan beforehand to collect a series of tissue sections from each sample. And then in the spatial multi-omics analysis case, you sort of, in a serial way, do one analysis, then do the next analysis, then do the next analysis. And so that's sort of what we're pointing out here on the slide is the first section was for H&E microscopy, the second for spatial transcriptomics, the third for spatial proteomics, the fourth for MSI. And so we sort of make this a template that can exist across your project in Weave. And this enables you to do bookkeeping and quickly set up these experiments because you're going to have, you know, let's say 15 samples with 4 modalities each. You're already looking at 60 data sets. So if you look here, we have samples, then within samples, we have a sample, then within a sample, we can create a stack. And then each of those come from typed data sets. So what that means is that anytime you put data on Weave, we have a type associated to it. So we understand it's an H&E or we understand it's spatial transcriptomics. The data doesn't just exist in a vacuum with a name on it. And then we have these slots and we say, you know, I have my H&E and everything's going to be registered to that. So I can create these registration paths so that I spatially align, for instance, what's in slot 4 to what's in slot 1. And you can set some specifics to that as well in terms of what kind of registration model you'd like to use. We offer automatic and manual non-rigid registration. And so that's sort of the idea of the stack template. So you define this once, this set of registrations to enable your spatial multi-omics experiment, and then you just repeat it, you fill it out across the different samples, and it keeps it organized for you. In terms of registration, so we do, like I mentioned, the non-rigid. So we have the automatic registration. So essentially you have pairs of images in the stack. One is fixed and one is moving. And in this case, our automatic will take those 2 images and find intensity features or just different parts of the data that you can select through parameterization to best perform the automatic alignment. But if that fails, we always have, you know, a second scenario, which is just using manual landmarks. So for instance, I have, you know, landmark one corresponds with this area between the two images. And so in this case, this is also a non-rigid registration, which means that it can account for localized warping, which you typically see when you're using serial sections for doing this kind of analysis. Within same section analyses, so if you have two spatial biology, spatial omics data sets from the same section, we can usually always achieve nucleus to nucleus registration. All right, so I'll move on from here. And so this is just an example. So this is an H&E. This is an example we did with MD Anderson Cancer Center in the US. So this is a MSI, post-MSI mass spectrometry imaging, H&E. And then we are able to overlay it from a second section with multiplex immunofluorescent. This is from the Luna-4 comet. And then we can also overlay in MSI. So one thing that I just note here is that our registration is capable of dealing with different fields of view. So for instance, a lot of cases, some of your instrumentation can only capture a small area of a tissue section, and that can make some of the registration challenging, but we handle all of that sort of in the background of all of the registrations. So in this case, this MSI and this registration, we're able to combine these three modalities into a single coordinate space for multi-omic integration. And this is just an example of, you know, H&E and multiplex immunofluorescence, again, COMET and H&E, and we can see overlay of the individual, in this case, stained by hematoxylin in the nuclei, and in the other case by DAPI. We can see the overlay of those two signals, which is expected because they both mark the nucleus. And then really the stack fusion that Maria mentioned, this is our idea that you have some sort of spatial reference. So for instance, a cell, or we also offer making arbitrary spatial references like tiles of hexagons or tiles of squares across a data set. And you can aggregate different measurements to those spatial footprints. So for instance, you can see, you know, what's my spatial transcript signal at a given cell, and then what is also the spatial proteomic signal at that same cell or area. And sort of this sort of becomes a data table that gets longer horizontally as we add more and more features to it. But that lets you do things like simple things like differential analysis between multiple features, or regression type analysis across multiple features as well. See if something in your proteomics correlates with something in your transcriptomics. And then layered on top of that, you also have things like spatial annotations made by a pathologist, or cell type annotations, whatever it may be. And this is really like a data frame sort of interface that we can access. And this is just an example from the previous registration example I showed. So in this case, we're aggregating to these mass spectrometry pixels, which are 20 microns by 20 microns. And then we have 5 elements that we're aggregating across the data. So we have some markers, so antibody markers from immunofluorescence, peptides, metabolites, and glycans measured by mass spectrometry, and then cell types as well, that are derived from the immunofluorescence. And this is just an idea of how we do this kind of aggregation. So we have one 20 micron by 20 micron area, but that obviously contains multiple cells. So we can do things like calculate what's the proportion of a particular cell type under this one spatial footprint. So in this case, you know, it's 60, 70% of this particular MSI pixel is covered by this particular cell type. And that's how we deal with multi-scale data in this case. But what does that mean in terms of results in the analysis? So in this case, we were looking at ovarian cancer, sort of different variants of ovarian cancer. and we were able to see across the sample sets that there are metabolites and glycans with sort of a differential correlation between our different disease states. So in these phenotypes that are MRD positive, we see a different correlation matrix than we do for MRD negative. And then we can go in and find out what do those individual metabolites and glycans in this case, where are they really different between the two disease states. And then obviously once we can also leverage the data that we get, that we can derive from one data set to drive analysis in a second data set. And what do I mean by that? So on the left, we're showing immunofluorescence data, and I'm showing the cell types that we've identified. So our collaborators are very interested in sort of the tumor and stroma interface and how these solid tumors change away from the interface. So we were looking at things like CAFs, cancer-associated fibroblasts, and immune cells, as well as tumor, which you can see here. So we can identify the cell types from one modality and use those cell types to drive analysis in a second modality. So in this case, we're seeing glycans, here, proteins. So we're seeing proteins that we can see in the tumor, as well as metabolites that we can all associate with the tumor that we're getting from a second modality. So with MSI alone, we couldn't really identify very finely these different cell types and associate them to the molecular signals because we simply don't have the resolution or the information from one modality to power that kind of analysis. And then obviously we're interested in spatial biology, so having layering and spatial information is really critical in that sort of analysis. So this is the same set of data. And what we're seeing here is that, as I mentioned, our collaborators were interested in the tumor and stroma interface and characterizing spatially what happens very close to that border. And so you can see here this, this color gradient from blue to red is going into the tumor or away from the tumor into the stroma. And we can characterize what are the cell types that we see at that border region. So for instance, we can see some cell types like CD3, CD4 T cells sort of at the right at the border in the stroma border, but as we move further away, they're no longer present. We can see some cell types that tend to really be right there at the border, but not in other places. So we can sort of look at this spatial profile of cell types in relation to different spatial structures. And yeah, we can do a lot of deeper visualization. So our platform's pretty generic at this point. So we can do simultaneous visualization of multiple modalities, whether it's proteomics, transcripts, mass spectrometry imaging. So that's what you're seeing here. This is a visualization we did with A-star showing different signals in COMET, so in multiplex immunofluorescence, combined with xenium data and showing an overlay of those. And then we can see, you know, you can find the same signals between the two modalities and compare them. As well as we've done a lot of drug work as well. So looking at drug localization in different mouse models. So this involves, again, we're looking at drug metabolites, combining that with spatial proteomics and spatial transcriptomics, which give us maybe a layout of like the morphology of what we're looking at, whereas the mass spectrometry imaging gives us really the actual detection of the drug metabolite in situ. So this is just some more examples of things we've done. Some more recent, research features we're getting into are studying multicellular environment analysis. I'm sure people who are working in the immune cell world understand very well that you have immune cells, but they're where they are, what their environment is also really critical. And so I'm just showing here in this visualization, we have sort of our standard UMAP embedding, this dimensionality reduction, which takes all of the different features of the data set, so all of the genes that are detected in this MERScope of a mouse brain section. We have all of those different genes, and then we have the cell types that are derived from those genes labeled on the UMAP plot. But what we can also do is that we can start to look at the cell type neighborhood. So for a given cell, we can say, what are all the cell types here? So we're no longer looking at features so what I mean by features are like molecular readouts at each cell. So what is the gene expression or the transcript count? But rather we're looking at what does the neighborhood of each of these cells look like? And then we get this second UMAP embedding, which has a lot of clustering to it as well, but not necessarily a clustering based on the cell types. But what we see is when we start to cluster this spatial embedding based solely on relationships between cell types, is that we start to reveal a lot of the morphology of the mouse brain that we didn't necessarily previously capture when just looking at cell types. And we think this will be interesting for finding spatial areas within tissue sections or small groups of cells that have particular patterning and across even multiple tissue sections. Another thing that we provide, and I'm almost wrapping up here, is that we provide standard spatial annotation tools. So this is just obviously marking up histology sections, but you can also mark up multiplex immunofluorescence. Any image that's in the platform, you can mark up as well, and keep all of this organized in a project-based way. The other thing that I don't have a video or anything, but we also have collaborative and interactive reporting. So basically every report you see in Weave in our platform is also, you're able to create comment threads. So I'm just showing this here on the right. So you can actually save the visualization that you were looking at any given time. This is what we call a snapshot. And then you can share that as a link to someone else and they can see exactly what you were looking at, as well as the title and description of what you were looking at. So you could, if you found something interesting, and we think this is really important for creating conversations between different people working on the same project. And they can not only see what you were looking at, they can also leave a comment about it. And it sort of like becomes a chat at that point where different people can in real time chat with each other about the visualizations they're seeing. And then finally, I won't go too into it, but one of the things that we enable is using our SDK, our software development kit. And really what this allows you to do is access data on Weave as if it were on your local computer. So you can do things like, for any computational people, you can basically access the data as Dask arrays or using ZAR or other common Python imaging libraries or ANData. So basically, it's like you're doing the analysis, but you haven't even downloaded the data to your computer. So that, and the other thing that enables you to do is you can access the data, run your analysis, and then push your results back onto the platform. And we have a bunch of convenience layers there that help you get it properly formatted before it goes back up so that it is immediately available in visualizations and doing other analyses that we already enable. So we really aim with these sorts of features to enable computational teams to integrate their workflows into our platform. And then we're eventually going to enable that in a deeper level. But at the moment, you can run them and then push your results back pretty easily. And with that, I thank you all for your time. And we'll be happy to answer questions at the end of the session. Thank you.
Okay, so thanks, Heath and Maria, very impressive. A lot of questions come to my mind, but let's do it at the end of the webinar. So now it's time for Eduardo Aranda.
Okay.
That is going to explain to us an example about his results in lung metastatic samples. I forget to mention in the introduction, but the title is so so specific that this is a computational webinar, we are not going to explain about different techniques or the wet lab techniques, but we have included here just 2 slides to show to you how we proceed with the samples before the sequencing. So, Edu.
Okay. Hello. Could you see
Thanks.
my pointer?
Yep.
Okay.
Yes.
Good afternoon, everyone. I'm Eduardo Aranda. I'm pharmacist and bioinformatician, and currently I'm doing a doctoral thesis in Dr. Rebeca Sanz research groups, Cancer Heterogeneity and Immunomics, in the Aragón Health Research Institute. And today I'm going to explain you and talk about the Decipher crosstalk between cancer and immunocells in lab metastatic samples using spatial transcriptomics. And also I will explain the techniques and highlight the key points analyzing this type of data. Okay. We are analyzing lung metastasis in colorectal cancer, and maybe you ask why lung metastasis is typically cancer colorectal metastasis to liver, because previous studies for our group suggested that the lung metastases are more immunogenic than the liver metastases, and also we observe here in the left in this picture that is the lung metastasis have also higher immunoprofile than other metastasis sites such as the brain and the bone. And we want to understand the intrinsic molecular interaction between tumor and immuno and stromal cells, because it will allow us to further understand the immunogenicity of lung metastasis and their influence on response to immunotherapy. Okay. For this purpose, we have analyzed samples from lung metastatic from cancer colorectal. And as we can see here in the right, this image shows our examples studied with hematoxylin and eosin. And during this presentation, I will show you some of these samples. There are several techniques that can be used to analyze transcriptomics, such as bulk RNA-seq, single-cell RNA-seq, and spatial RNA-seq. For example, with bulk RNA-seq, it's possible to measure the average gene expression across all cells in a sample, but we can know which cell types express each gene because all cellular signals are averaged together. And also, we don't have the spatial information. We lost this information, and we don't know where in the tissue the expression is coming from, which means that we miss the architecture and spatial patterns, and we can't see, for example, immunocell infiltration or detect intrasample heterogeneity, such as rare cell population. However, with single cell, it's possible to distinguish individual cell types and detect rare subpopulation. But it's not possible to have the spatial information. We know which gene are expressed by each cell, but not where in the tissue. And this we can know using a spatial transcriptomic. The importance of this technique is that we know where in the tissue each gene is expressed, and also this technique combines morphology with transcriptomics and allows us to detect patterns like immunocell infiltration, or for example, we can detect all the interaction between the tumor and the surrounding tissues, such as stromal or immune cell infiltration. Spatial transcriptomics is a deep dissection of RNA-seq data in a spatial dimension within its individual tissue slice. As we can see here, this is the histology of one sample, and here in the right is the histology with the gene expression of each spot. As we can see here, for example, this is a more purple region. It corresponds to the tumor region, and here are the spots in red. And the spots, or these points, refer to smart regions in the tissue where gene expression is measured and contains a mixture of cells. Each spot is approximately 55 micrometers, and you can see in each spot which gene is present and in which quantity. And for example, this sample has 2,229 spots. The workflow to analyze this type of data can be divided by 3 parts. The sample preparation, which include all the experimental parts performed in the laboratory, the sequencing, which include the preparation of libraries for the sequencing of all the transcripts in each spot, and finally the bioinformatician part, processing all the data, analyzing, and its subsequently visualization, which part is the part that I'm going to focus on today. One thing important is that these experiments have been developed using the technology on 10x. This special transcriptomics experiment is performed using the vision synthesis spatial gene expression for FFP platform for 10x genomics, and this use spatially barcodes, oligoprobes on a slide that hybridized to a poly-A tails of mRNA, capturing gene expression with the spatial context. First, the sample preparation, which is performed in the laboratory, is approximately 2 days to can perform all these steps that I'm going to explain now very briefly. Start with 5 micrometers of tissue sections that are put on the slides of the vision. For each, subsequently, the paraffinization and staining with hematoxylin and eosin and imaging. And this step is very important because to obtain in this step the images that will be aligned during the analysis. After that, libraries are constructed following different steps: the cross-linking and the pro-hybridization overnight. This is like the steps of the first day. And the second day, the following day, start with the washing and pro-obligation, and later we introduce the vision slides, this, in the vision synthesis. And here the probes are released to the vision slides. After that, the slides, the libraries are extended, eluted, and purified using the magnetic beams. And later, the last steps consist on the sample indexing and the last purification, resulting in the final libraries. Before sequencing, it's essential to perform a quality control of the libraries. To do so, we use 2 approaches here: the capillary electrophoresis, and here the real-time quantitative PCR. After that, it's possible to pull all the libraries and sequencing using the Illumina instrument and its protocols. This Illumina instrument generates a FastQC fields that can be running using the Spin Ranger pipeline, which is useful to align all the risks in a genome reference, and a count matrix is generated. This count matrix can be for the processor using Loop Browser here, or with other community tools such as Seurat, which is a package of R and it's widely used in many studies. Initially, the Spin Ranger also generates a summary report, as we can see here, with sequencing and processing metrics. And it's very useful to ensure that everything meets a set of minimum quality requirements, such as the number of rings or the number of detected genes in each spot, in the points, and also in all the tissue. Also, with Seurat, you can do more in-depth quality control, which I will discuss later. Okay, after the quality control, you see only the high quality spots. We can proceed with the clustering all the spots, but the expression, visualization, the genes of interest, and also we can evaluate all the tumor microenvironment. But now all these steps I will explain in more detail. Starting with the Spin Ranger. Spin Ranger is a collection of pipelines developed by 10x Genomics to process special transcriptomic data. It's recommended to run on a server because it needs a minimum of CPU cores and RAMs, as well as a large amount of memory and storage capacity to handle the data efficiently. There are different pipelines that can be used, but it depends on the type of data and analysis. For example, we have used the count pipeline because it's useful to analyze all the risks with a genome reference and obtain the count matrix. For example, MK-FASTQ is useful if you have the BCL files that are generated with the Illumina sequencing, but in our case, we have directly the FASTQ files. And with MK-RED, it's useful if you have to create a new genome reference. For example, if you add in the probe set new genes that you want to study or analyze. And the last is aggregation that allows you to combine sample for joint analysis, or for example, to do a cross-sample normalization is very useful, this pipeline. To run the Spain Ranger, also is very important to load a number of input, for example, the tissue imaging that is created with the microscopy that I named before in the laboratory experiment, the theta-6 imaging, the slide serial number and capture area, which is unique for each sample, the FASTQ files, the genome reference, and the probe set. And with all this input, you run the Spin Ranger on your server, and there are all of these output that are generated, that are a secondary analysis results, which includes information of the PCR and UMAP, the cell file, which can be loaded to analyze all in using Loop browser, a gene expression matrix that can be, for example, used to use Seurat or other package in R, the molecule level data that is the UMI information, read alignment files, which can be created if you want to create the BAM files, and the last is the summary report, which I named before. After run Spin Ranger, you can start to process all your data that can be analyzed using Loop Browse. That is a desktop tool for analysis and visualization developed by 10x Genomes, and also can run custom differential expression analysis. And later, a community develops tools that I said before, like R and Python and Seurat, which is a packet of R. It's the packet that I use to analyze all my samples. And why I use Seurat? Because allows for a more personalized and flexible analysis, being able to perform a higher quality control and filtering, and also allows to perform analysis with several samples at the same time. That is very important because with Loop browser, only you can analyze one sample and later other sample. And as well with Seurat, I can generate graphs that Loop browser does not allow, such as dot plots, for example. Later, after start with the processing the data, the quality control is maybe one of the most important steps, because with this step, you can remove low quality spots. For example, there are some spots that may contain little or degraded RNA, or maybe there are some spots that are outside of the tissue, or maybe a spot that there are middle of the spot outside and the other inside of the tissue. So they can contain not a lot of RNA. And other thing that is important is reduce technical noise, that sometimes this type of techniques can contain this type of problems. Other to avoid and prevent misleading interpretation, and also to improve the spatial resolution, because filtering ensure that only high confidence spots with strong expression are used for the other analysis, and also to enable accurate integration across samples.
Kako se vika na predavanje, ne se javim. I zato vikam, ajde bolje da imam.
Sorry, I follow.
Yeah, continue, Edu. Please, can you put the microphones off?
Okay.
Thank you.
And okay, for example, this could be important if you want to analyze multiple samples, and you can, with this, remove and minimize the batch effects. Okay, now I come. Okay, yes, I follow. This is a table that refers all the parameters that we have used to the filtration and analysis our samples. For example, here, all the spots whose percentage of expressed genes is higher than 15% in mitochondria have been removed. And why? Because they can indicate necrosis in the tumor, or maybe because they are with a degraded RNA. Later, the same parameter have been used to the ribosome, but also we have not eliminated any and removed any spot with this parameter. For hemoglobin, we have established 1% to remove region with blood that can also lead to a bias. And also we introduce NCONs and NF-future. NCONs is the number of genes detected in each spot, and NF-future is the number of genes, the unique genes detected in each spot. For example, here I can have one spot, but the NCON of I can have one gene, a unique gene, but this gene can appear more times, like 20 times, and this refers the NCON. And with these values, we want to remove these spots that are outside of the tissue, or maybe in the middle. Middle of the spot is in the tissue, and the other middle outside. And after all these parameters, this is the result of filter our spots. Before filter, we have this number of spots. And after filtering, we have this number of spots. And for example, here that refers the spot removes. After the filtering, we have for example, for one sample, and for other, we have a lot of more than 700. And it's depend on the sample, also depends on the size of each samples. Here I showed 3 examples of the result of this filtration and this quality control. For example, here we can see that these spots have been removed because have higher than 15% of mitochondrial genes, and is because there are necrosis or degraded RNA. This show the importance of the hemoglobin parameters. Here we can see a lot of blood, and here we can see the result of these removed spots, and it's because, for example, we can see that there is a broken tissue also. And the last, this is an example before and after filtering using all the parameters in one sample. It's a small samples because they don't have a lot of spots, but here we have removed 306 spots. As we can see here, there are some spots outside that have been removed. Another that are in the middle also have been removed because they are in the middle of broken tissue. Later, when we have a high quality spot, we can follow with the clustering analysis. To cluster our samples, we have used two approaches. Clustering according to the expression pattern, using a function from Seurat package, and another using estimate, that it's another package of R, but it's different to Seurat. And with this, we can classify our tissue according to immune, stromal, and tumor and immune cells. According to Seurat, one of the most important parameters is the resolution. As we can see here, figure resolution is less clusters, and with a higher resolution, there are more clusters. For example, in this sample, using 0.2 resolution, there are 5 clusters. With 0.8 resolution, we have 10 clusters in our samples, and with 1.5 resolution, we have 14 clusters. Here I show the results to 4 of our samples with a resolution of 0.2. As we can see here, for example, in the top of the slides, there are with a resolution of 0.2, there are 3 clusters in each sample. We can see in the UMAT, all the points are isolated. However, in the spatial resolution, here are separated all the clusters, but here we have the clusters together. There are some points of different clusters together. And here, for example, in this sample, we have 4 clusters and these 5 clusters. And now here I present the result using estimate, and that we use for clustering the results. I show the same sample that I use in Seurat, and here we can clearly observe the segmentation of the sample. Here in the left of the picture we have all the stromal region of our sample. However, in the right we have more all this tumor area that is surrounded with a tumor but with less score, and also this region is combined like a little of tumor and also with immune cell infiltration. Here I showed a result that with Seurat and with estimate we obtain more or less the same results. For example, here in Seurat the red points, these clusters correspond to here in estimate to the tumor. Later, the blue points here, this cluster correspond to all the stromal region of this sample, and also, and here, sorry, with all these clusters, the green points correspond to the also stromal score, but have also cell immune infiltration. Later, other thing that is important is visually the gene visualization. You can see all the genes that you want. And for example, I show here a very simple example using an extramal gene, the CD74 that is in the stromal region, and the EpCAM that is in the tumor region. That is an epithelial gene. And here also we can see in a violin plots, for example, the epithelial gene is in a tumor cluster is with a very high expression level. However, the stromal gene in this cluster is with a very low expression level. And here is another example. The same here, all the stromal region, and here the tumor. Okay, now and the last point that I'm going to speak is evaluate the tumor microenvironment. Here, one thing that we believe that is very important is to analyze all the hallmarks of the cancer in our sample. We analyze individually all the hallmarks, and here I put, for example, 2 examples. Here, the regulating cellular energetics. We can see here home is a tumor hallmarks. Home is very expressed in this region that correspond to the tumor. However, the tumor-promoting inflammation is a stromal hallmark of the cancer, as we can see here. And returning to the clustering analysis, one thing that we wanted to do to could compare all our samples is segment each sample into 5 clusters according to the stromal score that is generated with estimate. Being the cluster 1 and 2 here, the more orange points being the tumor regions. And here the greens correspond to all the stromal region. And these the 3 clusters that is in the middle between a tumor and cluster here correspond to this region that I said before, that is a little of tumor, a little of stroma, and cell immune cell infiltration, sorry. And why we did this? Because here we want to evaluate all the hallmarks of the cancer in our clusters. Here we have all the clusters, the 5 clusters per each sample, and all the samples. And here we have an amazing result because here we have the sample 4, the orange, the sample 8, and the green, the sample 5, that we classified before to do this as a low samples. These are with low samples according to the immunogenomic that I named before, and here the high immune samples that are the other 4. And as we can see here in the heat map, first our samples are divided between if they are high immune or low immune, and later are divided between stromal and tumor region. As we can see here, for example, in the high immune samples, we have the tumor samples are together here, and here the stromal samples. And for example, we can see that the regulating cellular energetics and non-mutational epigenetic reprogramming hallmarks of the cancer are more tumor hallmarks. And however, other hallmarks as avoiding immunodestruction, tumor promoting inflammation, activation, invasion, and metastasis, and inducing angiogenesis are hallmarks of the stromal region of our samples. And with this, we can compare all of our samples according to stroma or epithelial areas. And other tools that we use to evaluate the tumor microenvironment is use Tolkien. For that, we analyze in each cluster the genes that are differentially expressed with respect to the rest of the samples, and we have passed these genes to the Tolkien tool, which was co-developed by our group leader supervisor. This tool is very useful to visualize the molecular interaction between the clusters, as we can see here. For example, the cluster 0, cluster 1, all the cluster, and they have the receptor in triangle, and in circle the ligands. And this is very useful to evaluate all the communication between the tumor, the stromal, and immune cells between these clusters. Here I put an example in bigger that we can see. For example, here there are the Wnt phase A pathway, the non-canonical double Wnt pathway with his receptor, that is the Frislitz receptor, and his co-receptor that are ROR2 and ROR1. And for example, we can see this pathway that correspond to the ligand 4, the cluster 4 ligand, and is communicated, for example, with his co-receptor in with the receptor of the cluster 1 and the receptor in the cluster 3. And this is an example of pathway that, for example, is related to cell migration and inflammation. And in the last slide, we can see also how each cluster is communication and study the cell-cell communication. And for example, here in the cluster 0, the tumor cluster have a 9 interaction, for example, with the cluster 3. And here is a summary table with all the interaction between one cluster with the other cluster, and so if is statistically significant with the p-value, and with this we can see if indicate enrichment communication between these cell types. And the conclusion about this presentation is that spatial transcriptomics combines histology and gene expression. Either private or community development tools are very useful to analyze spatial transcriptomic data. However, with Seura, you can have a more flexible and personalized analysis. Also, quality control steps are very, very critical to remove this spot with very low quality. The result of expression analysis allow clear identification of tumor and stromal areas that overlap with the histological images, because this technique combine transcriptomic with the tissue image. And the last, spatial transcriptomics allows to study interaction between cancer cells and the tumor microenvironment, such as the stromal and the immune cells. And the last, I want to thank you for your attention to all, and thank my laboratory colleagues and also to all the collaborators that make this work possible. And finish.
Thanks, Eduardo. And also thanks to Maria and Heath. Well, thanks to the 3 speakers for a wonderful presentation, and also because they are exactly on time, exactly
To the minute, yes.
to the minute, yeah. So now we are plenty of time to discuss, to ask questions. So feel free to ask Eduardo, Heath, Maria, or even Maurizio and myself.
Meanwhile, I wanted to notice that the number of participants went up to over 90, so I'm very happy about that. And for those that maybe joined later, webinar will be available. It can be watched and rewatched as many times as you want. Will be on the website, available to the website.
Microphone on?
Yeah.
No.
That for question or no? Maybe I can start with a couple of curiosities. So, well, I can start with Edu about I think one of your slides you showed that an analysis where you combine the data from the multiple samples. So is that an easy task? Because actually the question is then also for Maria and Heath, like we've seen a lot in terms of exploring the single sample, quantifying, observing what's happening, and it's amazing, I mean, to navigate at that level. But how easy is to extract metrics, quantification, or information that you can look at across samples and use it for, further downstream analysis?
Yes, it's a little difficult sometimes to compare different samples because can have different gene expression. It depends on the tumor region or the site of the sample. But to compare all these samples, we divide each sample in 5 clusters. Whom I said in 2 clusters more tumor region and 2 clusters more stromal region. And with this, we want to compare all the sample. And for could remove the batch effect between cluster, we did a very in-depth quality control to can compare all these samples. I don't know if I asked your question.
Yeah,
or...
no, I saw, for example, that you use different thresholds for different samples. So, like in terms of like a minimum number of genes detected, did that actually help to make them more comparable or introduce differences?
In theory, the way in which we have calculated the score allow us to compare between samples, but we still have doubts.
Yeah, it's like
in theory,
learning process. Yeah.
In theory, it's like when you have a gene set enrichment, GSBA or something like this. And our positive control is that samples without stroma or with less stroma cluster with those non-stromal clusters in the high stromal samples. It's difficult to explain because we have samples and we have clusters. So we have tried to divide samples into the same number, samples into the same number of clusters.
Yeah.
Using stromal and epithelial markers. But actually, this is the question I would like to have to ask to Heath. How do you compare between samples? Because it's the same, Maurizio said. With these technologies, you have a powerful characterization of one sample that is cool, but we also want to compare across samples.
Well, yeah, I think what was shown by Eduardo is it's the same sort of case we'd use where we have some sort of batch normalization and QC step. I think that's essential. And when you have multiple analyses, it's also a question of multiple batch normalizations across those different modalities or within each modality. But yeah, like with these multi-sample studies, it's obviously very important to have an eye on what actually happened in the experiment, sort of have an eye on when all the experiments were run, so you can actually have some idea of where the batch effects may be coming from. If there's a systematic error, that's something you can detect with metadata in some cases. But yeah, so yeah, we have a typical QC pipeline using very similar tools to what Eduardo was talking about.
And Heath, can I continue? Maurizio, yes, right.
Sure.
Have you tried to analyze together spatial and single cell transcriptomics?
Yeah, so we haven't done it like a lot, but it is something that we are starting to do more of as we see collaborations that have this. So typically what we're seeing is like a subcellular spatial transcriptomics modality like Xenium or Cosmics, and then there's a single cell data set next to it. And you know, our technique has mostly just been to take the cell type annotations on the single cell data and try to transfer it to the Xenium data or whatever the other cells that we have from spatial, because they're usually like the single cell data is much richer in terms of transcript counts and it's richer data set, let's say.
Yeah, in theory, it is not necessary if you can achieve single cell resolution with spatial, but it's difficult.
But I think you still, I mean, we definitely see with some of the platforms like the Xenium or Cosmics or Merscope, I mean, NanoString now has like the whole transcriptome single cell, but the issue there is not like is still a detection issue. You don't have the same amount of detection that you have as you would in a traditional single cell experiment. So you get much lower transcript count in these single cell experiments. And that's probably just a matter of time, right? Like these techniques have been around for just a handful of years, so they still need more time to mature a little bit.
Great, thanks.
Yeah, in a way, like related. Yeah, there's a question from Theo.
Yeah, I have a different kind of question. Hello, everyone. You mentioned that this is the result of an EU program. If you can say a bit more how this, what kind of program was that, and whether you support open science and have access to data sets from different sources, fair enabled and things like that for this kind of, let's say, research.
Is this question directed to me? I'm not sure. I missed.
I'm not. Anyone who knows how the story of the EU program, perhaps. This came out of an European program. This initiative. Am I correct? This is what you said at the beginning.
You mean about the webinar in general, right?
Not the webinar.
Okay.
The platform. I mean the platform.
No, I think like within the platform is a private initiative, but like some of the studies they're working on is from an European project. I guess was the Glio match.
Okay.
So the actual experiment, yeah.
Mm-hmm.
I think there's a good point there on. Sorry.
It's a for-profit organization. This company. Do they support open science and have projects for the research community, joint research perhaps, or access to the tools?
I mean, I think we support working with academics through grant-funded projects, most definitely. And in terms of open science, our metadata and our ability to share data is compliant with fair practices. Yes.
And related to that, you were mentioning about the possibility to kind of plug into the your workflow some like access the data in a way that you can go on with the analysis with your own script and possibly like open source software.
Yeah, I see that you have the API and things like that. All right, nice.
Yeah.
Thank you.
Maybe like related to that, like the methods that you decided to implement in Weave, like I mean, what is the source for that? What has been method that have been published or like internally developed, and you know, like every day new tools come up, and it's a fast-moving picture. Like, how do you deal with that?
Yeah, that's a good question. So, I mean, for us, if a method is under permissive licensing, so that's typically like open source software can come in lots of different licenses. Some of them prevent commercial use, whereas others don't. So, our policy is if it's a permissive license, we can incorporate it in our platform. But it becomes a question of like picking and choosing what resources you have to put towards certain analyses. So for instance, a big thing that we deal with is cell segmentation. And that's not an area where we spend a lot of time ourselves developing methods, but we try to take the best that we can find from the field, as well as, you know, be open to supporting what our clients and collaborators would like to use. And that comes with like the flexibility of how we develop things. But for sure, like some things we developed internally, like a lot of the registration tooling. But some things we do use open source, like and like we're trying now to do things like take a library like Surat or ScanPy and completely map those to our platform so that you can do things like call any of those methods directly on our data. So if you have a familiar pipeline, it's already there. But yeah, but like definitely, I mean, we wouldn't, and as well as a lot of the data formats we use are also based on open source models. So we don't, we're not trying to lock things out in that way for sure.
Okay. I see there's a question in the chat from Anna. Very interesting presentation. I have two questions for Eduardo. The first is regarding the technical part of the estimate tool. How is the immune infiltration score calculated? Is there a list of genes for which a sort of enrichment is evaluated? And the second is regarding cell-cell communication between clusters. Is token list-based?
The estimate the immunoinfiltration score is calculated based on gene expression signatures and also with all the tumor and stromal region. And according, yes, it's a list of genes is with signatures. And according to the second questions, Talking, yes, it's a list-based tool that is very user-friendly to use. And if you want, and I can write the link on the chat and you can see, but it's very simple. You put, for example, the number, the cluster, and a list of genes that you want to analyze, and it's based on list-based tool. Yeah.
Yes, if by list-based you mean the input, yes, it is not necessary to give the software gene expression radius or p-values. It's just list of proteins or genes. It's also list-based in the sense that actually Tolkien is very easy to use, but also easy to compute. But because basically we have search in all databases with ligand receptor interactions, create a huge database, manually created, and just compare your list with our internal database and creates the protein-protein interaction.
I write you another link in the chat if you want to use the Tolkien.
You also can expand the network to see what happened downstream receptors, but it's very easy to use and to understand.
Okay, thank you very much. So
Thanks, Anna.
is really easy to use, like Tolkien, because I have the list. while in estimate, they will also do the estimate, will do the computation, also taking the gene expression data, right? So I imagine it will be a kind of mean.
Sure.
Okay.
Yes. Talking is web-based and estimate is an R package. So you need to compute a little bit, but it's very, very easy to use.
Okay, thank you, Rebecca.
You are welcome. And if you have further questions, you can contact with us as usual.
Yes, thank you.
Thanks, Anna.
The question from Sandra.
Hi, everyone.
Hello.
No.
Hi.
Well, congrats, everyone. It was very interesting. I have a question for Eduardo regarding the spot-based spatial analysis, because we are handling now with some spots, for example, in the boundaries between stroma and tumor. Did you find any spots that express incompatible gene expression signatures? Then how do you handle this? Like, you remove these spots, or you do like a score of if it's more stromal or tumoral? Like,
Yes,
That's
there
a very
are.
good question.
Yes,
because I'm
like-
having, like, some people in the group are having this type of
yes.
problems, even with a small spots, like more single cell base, and in your case it's big spots that have many cells inside, no?
Yes, we have the same problem, and for that we divide our samples in 5 clusters according to the stromal score, and we say that, for example, the first and the second clusters are the tumor clusters, the 4 and the 5 are the stromal cluster, and the third is a little in the middle, and we don't use to divide between stromal and epithelial region and tumor region.
Okay. Great.
And we have also tested the convolution methods. So we have spots, all the spots in the sample represented like in a pie chart. So we have the majority 100% being the same cell type, but we have these ones in the boundary with different cell types. We are working on that. We have not shown results, but we have working on that because of your question. Yeah.
Yes, we are like waiting the result of a single cell that we have developed, and it's, how say Rebecca, we divide later each spot like in a picture and say, for example, the 80% of the spots is tumor, and the other 15 is stroma, and 5 is cell immune cells, and
yeah, and that happens in more spots than we expected.
Yeah.
So we are now sequencing libraries with the same samples, adjacent slides, that we have prepared single cell libraries to try to use the same sample to the convolute.
Then you will use the single cell data for the spatial
Yes.
annotation.
Yes, because we have done this with public data, and we think it's not the same.
Yeah.
Great, interesting. Thank you.
We can share with you also the result when they come.
Yeah.
So there was a question in the chat. I see that Heath is answering that, but yeah, feel free to share with everyone. Also, the question was about the possibility to support federated data integration.
Yeah, I think well, there's maybe I have to understand fully the question, but I think the idea is that you have multiple sources of data or multiple databases, and you sort of want to have a central place that organizes communication between all of those separate organizations.
To
And so I think
communicate with the different data sources through the federated FAIR, actually, mostly, let's say, way. And you can use, not in your server, but you can conduct whatever research you can do. And the question is whether your platform can facilitate this kind of work.
Yeah, I think we'd have to look into how that would work. I'm not as sure about all the details of that personally. I don't necessarily manage these aspects of the company. But I can say from our experience with working in the GlioMatch project, I mean, we become a repository and an organization to help manage data across multiple collaborators. Whether we can deploy in different scenarios is really up to is a kind of on a case-by-case basis. But I think if that's something that's interesting, we'd be happy to discuss more in another call with the right people, let's say.
I was curious about like some of the challenges that you mentioned at the beginning. For example, when you have like a different section that have been analyzed, and so the registration and the matching, sometimes it's at single cell level, but sometimes it's not. So how is that process dealt, and what are the solutions?
Yeah, so I think it's a question that that our researchers, the people that we're collaborating with, they sort of have to define that questions for themselves, because if someone comes to us and wants help doing analysis and they're like, I want to do this single cell analysis across 2 serial sections, I mean, we have to be honest with them and say that's not possible because you're not really looking at the same cells. Right? If you really want it to be on the exact same cells, it has to be analyzed from the same tissue section. It's but what we can do, and what we often do, is our stack fusion method can sort of arbitrarily, let's say, create spatial footprints. So this is the idea where, let's say we have a square, we can draw squares of a certain micron spacing across the entire spatial coordinate system where things are registered, and then we can integrate to those squares. So it's a representation of multiple cells across multiple sections. But in some cases that can answer the research question, but it certainly will never be single cell at that point. But you can still do a lot of differential and correlative analysis working at that lower spatial resolution.
Okay, so if I understand, is there like a quite dense communication between the user and the company? I mean, like if I'm a customer and use the platform, is there a pretty deep interaction, or is like what was the strategy?
So that depends. So like with our product, like, so the thing I'm talking about here with doing these sort of spatial things, that's like a standard feature in the product. So someone who uses it has access to that. But I mean, I think you said earlier that this is a new field, so there's a lot of new things coming out. So obviously, like, we don't have 100% coverage of every possible analyses that exists in the field. So oftentimes there will be research questions, and that's where myself and members of my team will work on those things to answer those questions as they come, let's say.
Okay. And I mean, related to the absence of standard or consensus strategy and all of that, like, for example, even tasks like segmentation, as a user, like, how can I get an idea of what's the performance of the strategy, or like, is there any specific analytic or pre-analytic aspect that could completely change the quality of the results? Or
yeah, so I think one of the areas where we're very strong is obviously in communication and facilitating collaboration. But one thing that we can do very well is that we can build complex dashboards and visualizations that everyone can access and that no one needs to install software to run. So that includes things like cell segmentation. So we have dashboards for doing cell segmentation comparison, where you take two cell segmentations, you can look at the molecular readout between matched cells of both of those and really see and respond, and then take your next action. And I think that's something that's difficult, like with when you're working in larger groups with bioinformaticians, you sort of hand something off, you get back a PowerPoint or a PDF. Whereas like what we can give back to collaborators is like something where they can ask all the questions they want of the data without having to go through a third party. So that's how we really think that, like, okay, like there's still a case for a lot of like human in the loop type analyses, and involving people and doing some of these QCs is still really important, and facilitating that is where we see there's an advantage with what we can do.
Okay, yeah, very nice.
Do you collaborate with bioinformaticians or with wet labs, or both?
Usually both, actually. We see like obviously wet labs come in, but typically, I mean, we like working with bioinformaticians, because if here's the thing that comes up, like we have a research question, someone says, we'd like to do this process. Well, in order for us to get that process built, have a user interface, that's going to take more time. However, I can say, you can now do this in the SDK, and that's something I can communicate to a lot better to a bioinformatician than I can to someone in the wet lab to actually run the process through the code. And so having both is really critical. But I think like speaking with the wet lab people, you often get a better sense of maybe the purpose of the experiment and like what their real goal is. Whereas bioinformaticians can be your facilitator between the two teams in a lot of ways for us.
Yeah, thanks.
Yeah, makes sense.
Okay, more questions? Still 20 minutes to discuss.
Yeah,
No
exactly.
questions?
I had a question. I was very curious about the clustering interaction tool. I wonder if you guys could talk anything more about that, and like what that I guess any details that you can share.
Yeah, Eduardo has shared the link, so you are free to go and just play. There are several softwares in open source, Chat, for example, and others that are more rely on gene expression correlation. So we decided to do this, that is very, very, very simple. Just to list of genes and search if some of them are ligand and interactors, and just put together. It's very simple, intuitive, and the idea behind is very nice. Just to put together ligands and receptors from different clusters of expression. And it's also allowed to put till 6, if I remember well, list of genes or proteins, whatever you prefer. Because, for example, in spatial transcriptomics, you don't have just 2 clusters, but many. So it's possible to see interaction between different clusters. And is the user is the person who prepared the list. So the output depends on the input, on your idea. You prepare the list differently in depends of your question. So it's easy to use. but you have to put in the input exactly the genes you want to know if interacts or not.
And so in this case, is it like, okay, I have two clusters and I have some differential analysis, so I've identified the genes
Yeah.
that are higher in both of those, and then I want to just see if there's some ligand receptor. Is that the idea? Like, how do
Yes.
you get to that list of genes that you want to input, I guess?
And also they perform a permutation test to see if the number of interactions you find are by chance or more than expected, to give you a p-value, just to give you an idea about how the interactions are by chance or really these clusters are speaking.
Interacting.
And the way you obtain the list is up to you. For example, the classical one, as you say, is differentially expressed genes between two clusters, but you can choose if one cluster versus all clusters, one cluster versus the adjacent cluster, wherever you prefer, whatever your question is. Yeah. Also perform enrichment analysis, downstream analysis, also search for drugs, blocking the receptors, just to give more information to the user. But the core is the protein-protein interaction network based on ligand receptor interactions, mined from literature and public databases.
Thank you.
Yeah, you are.
There is a question from Theo.
Yeah, since we have time, I would like to ask if, let's say, if I understood well, you support, let's say, the different teams with algorithms, insights, and things like that. Is that correct? You direct them.
Well, no, I mean, they can be self-directed as well. I mean, but we can help in a service capacity as well for difficult or challenging problems. We can help support those. But for routine things, like we think that our users can use the software and manage it themselves.
No, I mean, do you train the models together? And the question actually is, if an IP comes out of this work, do you share or some of these rights or what?
So for IP, I mean, that's something we negotiate with clients at the time of contracting. So usually processes that are developed, like usually we don't keep any data IP rights ourselves. But certainly like if we were to develop a process internally based on the data, but doesn't require the data, we would retain rights to that. That's just like the global view.
And related to the service part, not the software, but the access to a service possibility, how does it work? Like, what's the general scheme for that?
All right, let me do my sales. Try to be this as best as I
Might
can.
not be
I don't do sales. I'm not but typically we have like a license fee. That's typical access to Weave, and then services are additions to that. Is generally how it works. And the license is structured. It just is not on a per user basis. It's like a use basis. So like if you have a team, the idea is that everyone on your team can access the platform, so you don't have to pay per person. But yeah, that's and then usage comes from like if you do, it costs more to run 10,000 samples than it does to run 100, for instance.
So the service is actually like some additional, like if there's like additional interaction, But it's not like, I don't know, I have my data, I send you the data, I get back the analysis.
I mean, that can definitely, it just depends. Yeah, I mean, that's something that we have done that in the past. But typically, if you send us the data and we were to do something with it, it's the endpoint is going to be something you access on the platform, something you see, reports you see there. So it's not going to be just a file we send back. So
Yeah.
there'll always be some, like that's how we work internally, right? We use our own tools to do our own work, to do these sort of projects. So that's the way we communicate it communicate the results.
Okay.
Okay. No more questions. Not in the chat.
Quite a few people
Yes.
sharing
Living.
good positive thoughts about the webinar. So yeah, happy about that. And most of all, thank you to the presenters and yeah, for very clear and interesting presentation, I would say.
Yes. Thanks so much, all of you.
Thank
And
you.
thanks also to people to assist.
Okay, so maybe we'll think of additional or continuing the series with, since it seems there's quite a lot of interest on these topics and we have to and really fitting with the goal of IMMUNO-model. And by the way, if there are like here people that are not already part of the cost action, like I would suggest to join because it's a very rich and growing community of people interested in many aspects of immunotherapy. And actually, the computational part has been growing, and there's quite a high number of computational biologists that, in the last year have been interacting in many ways. So, yeah, for anyone that has interest in also in the computational part, I think it's a nice network to be part of.
Yeah, I agree. And also there are a lot of different technologies in proteomics. Perhaps we are now more focused on transcriptomics, but as Heath explained, also proteomics. Many people are using these new platforms and techniques, so okay, we can think about it.
Exactly.
Let's see the people if they want.
Thank you, Rebecca and Maurizio, for organizing this very exciting webinar. So I think we can close the webinar. So I will stop the recording.